All right, welcome. Today, we're cracking open a case file on some of the world's most slippery criminals, the ones we literally can't see. We're about to become microbe detectives, diving into the incredible world of microbiology to see how scientists identify, fight, and track these invisible threats. So go ahead and grab your mental magnifying glass. Let's get this investigation started. I mean, that's the million-dollar question, isn't it? When an outbreak hits, it's basically a crime scene, but with no fingerprints, no weapon, nothing you can see with the naked eye. The culprits are microscopic, but their impact can be massive. So how in the world do you hunt them down? Well, that's the mystery we're going to unravel today.
You know, every good investigation starts with a lineup of the usual suspects. So first things first, we need to get to know the main crews of microscopic culprits that our public health detectives are always keeping an eye on. Let's meet this microscopic rogues gallery.
Okay, so what's wild about the fungi family is just how diverse it is. They're not all bad guys. You've got Saccharomyces, which is basically a helpful civilian that gives us awesome stuff like bread and wine. But then you've got its cousins, like Candida albicans, which is the most common fungal troublemaker out there, causing infections like thrush. And others, like Aspergillus, can cause really serious lung infections. It's a classic case of a family with a few very, very bad apples.
All right, next up in the lineup, we've got some bigger suspects, the parasitic worms, or helminths. They're generally split into three gangs. You've got the trematodes, also called flukes, which can cause some pretty devastating organ damage. Then there are the cestodes, tapeworms, and these things look menacing, with a head full of hooks and suckers just designed to latch on and not let go. And finally, the nematodes, a group that includes some very common criminals like the human roundworm, hookworm, and pinworm. Now, the protozoa, these are single-celled powerhouses of disease. Seriously, this lineup has some of the most wanted pathogens on the entire planet. You've got everything from Giardia, a top intestinal parasite right here in North America, all the way to the infamous Plasmodium, the culprit behind malaria, which is the second biggest pathogenic killer in the entire world. These tiny organisms have an absolutely massive global impact. So what makes these protozoa so sneaky and resilient? Well, it's their clever survival strategy. They can exist in two different forms. First, there's the trophozoite stage. Think of this as the suspect actively committing the crime. It's out there, feeding, multiplying, causing trouble. But when things get tough, it can transform into a cyst. This is its dormant, protected hideout, just laying low, waiting for the perfect moment to infect a new host.
Okay, so now that we know our suspects, how do we actually take them down? This is where the forensics lab comes into play. Let's take a look at the tools our microbe detectives use to find the culprit's weaknesses.
This right here is a classic interrogation technique for bacteria. It's called the Kirby-Bauer test. So first, you make what's called a lawn of the suspect bacteria. You just spread it all over a petri dish. Then you place these little paper discs on top, and each one is soaked in a different antibiotic. You let it incubate for a bit, and when you come back, you can literally see which weapons were effective. And this is what it looks like. See that clear ring around the disc? That's what we call the zone of inhibition. It's basically a kill zone where the antibiotic has seeped into the plate and just stopped the bacteria cold. It's super simple. The bigger the clear zone, the more powerful that antibiotic is against that specific bad guy. So the lab results then get sorted into these three categories. S stands for susceptible. That means the antibiotic works. It's a direct hit. R is for resistant, which means it's totally useless. The bacteria literally just shrugs it off. And I for intermediate, that's kind of a backup plan. This info is absolutely vital for doctors so they can prescribe the right medicine and win the fight.
Knowing your enemy's weakness is one thing, but knowing the size of their army, that's a whole other ballgame. So next up, we need to figure out just how many of these microscopic bad guys we're actually up against.
Imagine trying to count every single bacteria in a sample. It's impossible. There could be millions. So scientists use this really clever technique called serial dilution. You take a sample, you dilute it. Then you take a bit of that sample, and you dilute it again. You just keep repeating. By systematically watering it down, you eventually get a sample that's thin enough to where you can actually grow it on a plate and count the individual colonies. And when you do the math and work backwards, the numbers are just staggering. For example, if you count just 112 little colonies on a plate that was diluted 10,000 times, that means the original single milliliter of fluid had over 1 million bacteria in it capable of forming colonies. It's just a powerful reminder of the incredible scale of this invisible world all around us.
Our detectives also need to know how fast the enemy's multiplying. And to figure that out, they create what's called a growth curve. It's pretty straightforward. You just take samples at regular intervals, say every 30 minutes, and you measure how dense the bacteria population is. When you plot that data, you can see just how quickly their numbers are exploding, which helps predict how severe an infection might become. So, we've profiled the suspects, we've found their weaknesses, and we've counted the numbers. Now it's time to zoom out to the big picture. How does all this detailed forensic work help us track the spread of a disease and solve an entire outbreak? When you're tracking an outbreak, our public health detectives, epidemiologists, they live and breathe these two key stats. You see them all the time from the CDC. Morbidity is simply the number of people who are sick. Mortality is the number of people who have died. And telling the two apart is absolutely critical for understanding the real scope of a disease. You've probably heard this term R0 a lot lately. It's a really simple but incredibly powerful number. All it means is this. On average, how many people will one sick person infect? If R0 is 2, one person infects 2 others. If it's less than 1, the outbreak eventually dies out. But if it's more than 1, it grows exponentially. This one number can drive huge public health decisions because it tells us if we're winning or losing the battle.
And in any outbreak investigation, the holy grail, the thing everyone is looking for, is the index case. You've probably heard it called patient 0. This is the very first person who got sick. Finding them is like finding where a fire started. It lets detectives trace the exact path the disease took and understand precisely how it spread through a community.
To show how this works, there's this great classroom experiment. One student is secretly given a piece of candy that's been coated with a totally harmless bacteria. Then everyone in the class mingles, shakes hands, whatever. Afterwards, they all swab their hands to see who got infected. By tracing it all back, they can figure out who the original candy holder, the index case, actually was. It's such a simple but brilliant way to show how epidemiology works in the real world. So there you have it. From checking out a rogues gallery of suspects, to using advanced forensic tools in the lab, to tracking a disease's spread across whole populations, the work of a microbe detective is this constant, fascinating battle against an invisible world. It really makes you wonder, with this incredible toolkit we have at our disposal, what microscopic mystery are we going to solve next?